Should I click something?
Just enclose. Okay.
So welcome everyone. It's a great pleasure for me to introduce today's speaker, Laura Alvarez. She studied chemistry in Spain, and after she started a PhD in Bordeaux, working with liquid crystal and particles. Then you spent quite some time in Zurich in Rujo Isada's group, working on active colloids. And she really did a very nice work trying to give some intelligence to this colloid, not just to observing their passive motion, but try to pilot the way they can move and navigate in fluids. And then you got a chair junior position again in Bordeaux, where you implement these ideas, maybe on a larger scale, towards self-mimetic microdevices.
Mm-hmm.
So the stage is yours, Laura. I'm looking forward to your talk.
Thank you so much, Antonio. I hope you can hear me well. Yeah, so it's a real pleasure to be here today and talk a little bit of the work that we're doing in Bordeaux, but I will also talk a little bit about part of the postdoc work, which is organically follows, it's kind of the predecessor of what I'm doing now. And yeah, so as Antonio said, I used to be a chemist. Now I'm more like a physical chemist, soft matter physics physicist. And today I will try to orient this talk from a more material science and physicochemical perspective. And I will be talking about how can we design self-mimetic microdevices using soft and responsive materials. And so if we take the approach of a material scientist and we try to look at robots and devices in general, we know that we are very quite advanced on robotics and devices at the macro scale, but there is now a focus on downscaling these units to the micro scale and specifically to the size of a cell. But it's not trivial. It's a quite problematic topic because we cannot just take electronics and start making nanoscale and microscale approach that we take in the macro scale. Of course, there's also some research on that, but it's quite difficult. So here, instead of just taking the approach that people do for generating microscopic robots, what we are thinking about in the active matter and soft matter physics domain, it's instead of looking at this, start looking at nature, right? Because cells are perfect machines. And I like always to show this video. This is a T cell, a neutrophil, so a white blood cell. And as you can see here, this white blood cell, it's swimming around, it's actually chasing this bacterium over here. And you can see how it's able to chase this bacterium by smelling chemical cues, by chemotaxis, reconfigure through this very complex environment, and finally phagocyte bacterium and kill it, right? And this is possible because these units, biological units, have evolved through millions of years, and they're very complex. The machinery that they use is quite complex. And if we want to reproduce this from a more material science perspective, we need to start breaking down all of these layers of complexity and start looking from a physical-chemical perspective, what are the different layers, right? So for example, we can look at the mechanical properties of the membrane of these cells. We can also start looking at how they move, their motility, and try to design this. But also we can go a bit farther and look into the chemical tools that they use to communicate or to chase bacterium, for example, and how all of this builds up towards a functionality. So we can use synthetic and biological units towards these micro devices, right? And that's kind of the overall objective of the field that I'm working right now. So this is a picture that is a bit sci-fi oriented, but one of the main goals is to develop these micro robots that are functional and autonomous, but also that we can also generate artificial cells. So units, again, that have functionalities and they look pretty much like cells. And so, of course, if we take again the picture of this white blood cell, it's very complex. But then in the lab, we can work with much simpler units to try to understand some of the fundamental physics and physicochemical properties. And today I will mainly focus on motility, right? This is one of the aspects that I showed you before. And so how can we reproduce motility? Maybe you have already heard about active particles, but basically we can use colloidal particles that are active, so they are able to consume an external source of energy into net propulsion. And as an any kind of active particle. In water, they exhibit Brownian motion. They can take different shapes like spheres. You can make these geometries by combining different spheres. We can make rods. And again, they need to be asymmetric. So there has to be an asymmetry on their shape or material properties in order to transform this energy into net propulsion. And so there's many ways of doing that. Probably you have heard also about catalytic Janus particles. So these are able to consume a chemical from in solution. And because of chemically creating this chemical reaction, there is a chemical gradient on the surface of a particle just in one side that creates an osmotic pressure. And there is a net force that pushes the particle into with a velocity that depends on the concentration of the molecule that is being catalyzed. And this process is called self-diffusophoresis. But there's other ways of activating or make particles self-propel. Mainly, we're working in my lab with external electric fields. And in this case, we take advantage of the difference in the electric properties around the particle surface. And what happens here basically is when you have, imagine that you have two electrodes, we apply an oscillating electric field with a given frequency, and you will basically polarize the charges on the surface of this particle in both sides. And because these timescales of polarization at both sides are different, you start creating electrohydrodynamic flows that are imbalanced. And because these are imbalanced, the particle will be pushed again through a net force in 2D. And this is a video of these particles. So basically, these are activated by electric fields, and they swim on the surface. And the velocity of these particles increase linearly with the square of the applied field. And you see that time to time they reorient. So this reorientation, it's just given by random Brownian motion, random noise in solution, and depends mainly on the size of the particle. And so, I mean, these particles are very versatile, of course, and people have been studying them for many years now, probably more than a decade. And so we know that when it comes to, if we want to start observing different motility patterns, we can play with different properties. For example, we know the dynamics depend on shape. Again, I showed you this video of the spherical particles, but now if we start playing with shapes, in this case will be like these L shapes that exhibit this chiral motion because they experience a torque. Now you can have another type of pattern. We can also play with interactions because in nature we also have interactions, right? They are not, biological units are not just individualists. So people also have explore how by tuning interactions, particles will explore the environment differently. And finally, of course, we always aim to have a functionality, and people have worked a little bit on cargo release, again, with electric fields by trapping particles and then release the particles. And it's a pity I don't have this video here, but people have also tried to create these cell bots or like sperm bots by just coupling a magnetic responsive helix with a lazy sperm, but that is still functional, and directed this to the opposite. So now you can see that there is quite room for improvement and play around with these particles. But at the same time, they also come with some drawbacks. So most of these cases, they have fixed geometries. And of course, as I said before, geometry affects dynamics. So if you have a fixed geometry, the dynamics will be always the same. They're usually activated by external fields, and they are not adaptive. So much of the effort that we are trying to do is basically to have something that is a bit more similar to a cell, because right now these models diverge from what we think of a cell and go towards autonomous and adaptive. And so today I will be presenting, as I said before, a part of the things that I was doing during my postdoc at ETH on these smart colloids, but then I will move towards what we're doing now in my group in Bordeaux. And so the first part deals with make active colloids adaptive. And so I'm asking you now, how can you make a colloid adaptive? Based that in, for example, if you think about dynamics and shape are related. Yeah?
I would say give it to the maximal freedom with respect to the geometry of energy pumping.
Okay.
So
yeah.
I don't break any symmetry. I let it do it by itself.
Well, actually, it's the opposite. You need to break even or change the
asymmetry.
Oh, okay, cool. Then we will talk. By the way, you can ask questions during the seminar if you want. Well, in our case, actually, what we try to do is to insert a sensor, a particle that's it's responsive to external stimuli. And I'll talk in a bit what type of material, but the idea is that we have our hard colloidal sphere, right? So now the symmetry is not only on the sphere, but we create this dumble, what we call a dumble particle. So this particle now, one is close to the other one, it's asymmetric. And then we have this polynipine microgel. So polynipine microgels are basically these spheres made of polymer called polynipine that is thermoresponsive and has a critical phase transition temperature, which depending on the composition is around 32 degrees. And so basically by crossing this phase transition temperature, the polymer hydrophobic interactions are quite high. It reconfigures, expels the water, it's kind of a sponge, if you like, expels the water in solution, and the size, the hydrodynamic radius can reduce its size by 50%. And so based on that, what we wanted to do is have basically this polymer, this microgel that is attached to the particle, and by increasing temperature through light, by changing light intensity, we will basically transition between these two configurations. And you can see that here the microgel becomes much smaller, so we are kind of affecting this asymmetry by changing the geometry of the particle. And I think in this video you can see that. So this big particle that lights up is the pink sphere, and by shining light and increasing temperature, I hope that you can see this is the blob here is microgel, and I hope that you can see that this is real-time video. It's quite fast process, so we can basically change the configuration, the geometry of this particle. But again, okay, we have the reconfiguration, we have this sensor that is now in the particle, and the question is this active, right? So in order to activate this, we will use, again, electric fields. So I will go a little bit more in the details of how this mechanism works. And so what happens here is that, again, when we apply an AC electric field with a given frequency, usually it's a kilohertz, we have these hydrodynamic flows arising in both sides of the particle. We can measure these flows by PIV, just by putting particles tracers that give us kind of the idea of the fluid flow profile. And so what we have seen is that at room temperature, like cold temperature, let's call it this way, blue, the hydrodynamic flow on the side of the microgel is dominant in the sense that this particle will move with this pushing with the pink side in front. And then when we increase the temperature, we actually have an inversion, because I will explain in a minute. So we have an inversion, so this microgel flow reduces its magnitude, but also the direction. And now the particle will swim, I think it's, yeah, on the other direction. And this is because the magnitude and direction of these hydrodynamic flows depends on this expression, but mainly on the Clausius-Mosotty factor, which gives you an idea of the polarization properties of the surface charges with respect to the medium, saying that what matters here is the surface conductivity of the particle. And if you imagine that surface conductivity is affected by like the charges around the particle, and you are decreasing the size of this, you will be also affecting the surface conductivity of this microgel. And we actually can measure this very nicely, this surface conductivity, by doing the electric spectroscopy, and we kind of feed the data to this expression, and it was supporting very nicely what we see experimentally. So, okay, let's see what happens then. So we have our particle, we apply electric field, we have our blue particle with a given dynamics. We increase the temperature by increasing light intensity, and at some point you will see that there is like a relaxation time, and this particle will start swim much, much slower. So not only the direction changes, but also the velocity reduces and the rotational diffusion increases because we're heating up the particle. And so when we switch back the light conditions to the initial conditions, so cold state, you will see that the trajectory relaxes back to the initial state. So this is a very reversible process that we can do many times, as much as this experiment holds the electric field, let's say. And here is basically the determination, or like the quantification of the velocity as a function of time. So you can see that indeed when we are at cold state, the particle has a given velocity. When we increase light and temperature, we have a transition state, and the velocity decreases by twofold, and then we can cool back again, and this will increase. And again, this is reversible. And what is very nice that hasn't been done before is that this is orthogonal to the propulsion mechanism. So we do have an external field that propels the particle, but then the adaptivity of the particle, it's independent to that. It's only because it's able to feel a temperature change in the environment. And so one thing that we can do with this, I don't know if you are aware, but the Di Leonardo in Italy, they have this nice bacteria, light-activated bacteria, where they can play with shapes and draw Mona Lisa. So our idea was also draw Mona Lisa with our particles, but well, it didn't work. But you can see it's not as efficient as nature, as bacteria. But what we wanted to demonstrate is that because we can control dynamics through light or temperature, we can now start working with local patterns or local local light patterns. And so what you have here is basically an initial sample where particles start to move. Then we switch on light only in this area here, and you can see that there is an effective accumulation of particles that start to swim slower and stay there. And then again, this we can do many cycles until the experiment is done. And recently we have, a PhD that continued this work. We have also worked on different type of configuration. Now I talked before about L shapes, so we can also construct these L shapes that have a chirality. And I will let you see the video, and I wanted to tell me what, apart from the change of radius of this chiral motion, do you see any other change here? From the blue to the red.
Deepka told me that they are making circles in the blue.
Yeah, in the red too. It's just that the radius changes, but there is something else. That too, but the chirality is inverted. So we actually invert chirality because the same way that before I told you, so you have to imagine that now the force, the propulsion force comes here in this unit. You have torque because it's not symmetric in that axis too. But we also keep this inversion of motion, right, that I showed you before and that I explained before. So basically we can actually tune the chirality of the trajectory on demand, basically. This is very nice because people now are very obsessed with synchronization of active matter. So we could play with this property. And then, you can also think of playing with different microgels, with different transition temperatures to play with state dynamics, basically. This is the work that has been published recently by Stephen. And so, of course, this is pretty cool, but something that we are also I was very interested not only in the responsiveness, but also in the collective behavior, right? As I said before, particles active, biological systems are not individualists. We are not either, actually. We do like to socialize, most of us. And so the idea was to reproduce at some point collective behavior and understand how can we use this from a material perspective or like soft matter application. And so, this is not something that I came up with, right? People have tried to reproduce this, as I said before, with different type of active particles, playing, for example, with dipolar interactions. When you have these Janus particles with a metal cap and you apply an electric field, you start to have this formation of chains at given frequencies. In the group of Denis Barthelot, for sure you know this work, the Quincke rollers, where they are able to in this case, it's not a Janus particle, it's just a sphere that they are able to make roll on the surface, and they create this very nice swarming behavior in confined systems by very, very important hydrodynamic interactions. And then you also have the formation of these clusters, what we call also motility-induced phase separation clusters, where basically what happens here, particles are moving around, they actually feel attracted to each other by also hydrodynamic interactions, and they form these nice clusters that become much smaller, and you have kind of these two phases where you have some sort of gas state and solid glass state, basically. And these are mainly due with self-phoretic interactions, where we have catalytic particles. But all of these works are only mainly dealing with one particle activity. So a particle that just has a different, distinct velocity. But again, if you look at biological systems, even us in marathons, there are not just one distribution of velocities of people running, right? We have a distribution of people with different velocities running fast and slow. Also happens in social behavior in ants, for example. And here what happens is that fast ants will phase separate from slow ants. And this question has been kind of explored by mixing active and passive particles, trying to mimic a bit what happened in these MIPS states, in which what they found is basically that the passive particles usually form these clusters. So the passive are the blue particles, the active particles are the red. And what happens is basically the only ones that move around and kind of break down from this cluster are the active particles, and that's it. So our question was like, okay, what happens now if we mix two particle motilities. So what happens if we have active particles that have two motility populations, so fast and slow? Can we play with this? Can we tune the particle interactions? Can we create a new state of collective behavior? And this has been now a long collaboration. I hope that this work comes out soon with a group of Ignacio Pagona Raba in Barcelona, Demian, and their former PhD, Elena. And so we were trying to explore this question. And so experimentally, how we do this is again with electric fields. It's kind of the driving mechanism that I know the most. And so what we did here is now engineer particles with two motilities by changing the coating or one of the hemispheres of the particle. So now what we use here are silica particles, and we coat them with a metal. But in order to change the motilities, we need to use two different metals. And so what we get here is that after a lot of trial and error, what we got is that if you coat a particle so this silica particle has the same size, around 2 micrometers, and you coat one with gold, the other one with palladium, turns out that the palladium particles are super fast at the conditions, field conditions that we're using. I mean, these particles move around 100 micrometers per second. They're quite fast, and this one a bit slower, but still very fast. And so again, we identify the propulsion mechanism by doing PIV, so recognizing how these hydrodynamic flows profile look around. And what we found is basically that the combination of these hydrodynamic flows around the palladium were the proper ones to make this particle faster. And so basically, what we do here is quantify the velocity as a function of frequency. Now, frequency here will be very important for later. And again, what we found is that, yeah, we have a population of fast particles. The slow particles, they are around twofold. And just to make a point here, this velocity should be frequency dependent. I mean, we see a slight change, maybe in the slow particles is higher, but in this regime, it's not such a big change, okay, within the error bars, I would say. And so this is what we did after is just calculate the Peclet number of these two particles just to characterize, it's just the ratio between Brownian noise and velocity of these particles. And so we indeed found that we have one of the populations that is almost twofold faster than the other ones. So that's great. We have our two populations of particles. One is fast, one is slow. But then we now need to go to this more concentrated or like dense system in order to look for collective effects. So, the frequency, it's a kilohertz. Yeah, thanks. Yeah, sorry about that. Yeah, it's kilohertz. So we are always working on the kilohertz range. I mean, people are actually working more in the megahertz, but the propulsion mechanism is different, basically. And so what happens now is that we take our particles. So here I'm going to show you just one population of particles, not the two at the same time. And at a given frequency, we just have something boring, I would say. Well, you have a lot of particles, but they're just swimming around. Nothing really interesting happens. So the colors here are the velocity orientation. Okay. Yeah.
Because if I understand correctly, the mechanism is controlled by the recirculation motion of the liquid around the particle.
Exactly.
How close can they get without interfering with each other?
I'm going to just talk about it now. Yeah, because so at this point, they do get close. I mean, I think that the distance of the particle distance, it's the radius plus, like, the d-by length, a little bit more than the d-by length. So that's what they get here at this frequency. But now if we change the frequency, we will change a little bit these flows. They will become more important. And what we have, I should accelerate this movie because it's more graphic, it's better. But I hope that you can see the reference. Right now we have interactions among the particles. They start to swarm in this very nice, what we call dynamic polar cluster state. It's not the clusters that I showed before where they just stay together. They are actually very dynamic, and it's very hard to measure cluster size here because we even have, sometimes it looks a bit percolation pattern. Yeah, but it's not. At the end, the large scale is not exactly that. But so yeah, basically what we have, or what the hypothesis behind is that we have a shorter range attractive interaction because these flows at lower frequencies become a bit more important. And then we have also alignment. And so, yeah, we have this transition from disorder to polar cluster. And what we try to motivate here now is that you have to mind that when two particles are coming close together at 1 kilohertz, sorry, they come close together, and because of these flows are attractive on the side of the silica particle, or the silica hemisphere, there is a short-range interaction that will keep the particles together for a little bit, the enough time for then reorient due to dipolar interactions between the metallic caps. And so they will align, they will swim together for a little bit, and then they scatter. So we have characterized this individually. And then you can imagine that when you have many of them, they will kind of start to get trapped in these clusters until the cluster just basically breaks. And in simulations with a Bishek model in ABP particle simulations, they kind of actually found this coupling parameter, or like alignment parameter, that just, it's a combination of like the competition between what will be noise and, oops, spoiler, noise and alignment force. So basically they can actually plug an alignment force parameter that will be very nicely reproduce our experiments with these worms. So this is for one type of particle, one type of either the fast or the slow. They actually behave more or less the same. So we have built a phase diagram for different frequencies and concentrations, and we always find at a given frequency and concentration this type of behavior. But what happens now if we mix slow and fast? And I'm really sorry if there is any colorblind people in the room. This was not the most smart choice. Yeah, I'm sorry about that. We will have to change this. But so if you see the color difference, so the fast are the red particles and the green are the slow particles. And I'm going to play the movie. I mean, these are the experiments. In simulations, it's better. I think you see what happens better. But I want to play the movie, and I'll let you tell me what you see. So again, red fast, green slow. Do you see anything arising from here? And the arrows are the velocity vector. That good. We also. So again, we have these swarms, which is nice. So now we have, but I hope that you can see that red, an average, phase separate with red and green with green. And in the experiments, you cannot see it very well. I mean, I can see it because now I have been through these movies like a lot. But now if we do the same, if we apply the same in simulations, we actually see that the red at some points if we take screenshots, the red will push, so the fast particles will push the slow particles. And this we can actually measure from the experiment. So these are experimental data. We can measure the probability distribution of displacements for fast and slow particles. And so basically in the dashed lines are each of the samples separately. So we have a distant distribution of displacement. But we see that when we mix them, so the slow particles become a bit faster and the faster particles become a bit slower. And of course, the slow particles are pushed, right? They are kind of like being displaced faster in space. And we can also, I told you that red B with red, green with green. We can actually look at the GeoFAR of, so basically global, so the whole sample. And then we can look interspecies, which is the interspecies, which is the light pink curve, which this means at the end of the day that basically red really like to be with red and green like to be with green. And here what we find is what we motivated that we find this cooperative dynamics in this very nice polar cluster state that people have not explored yet. Yeah, so I don't know if you have any questions on this. Yeah.
Can you tell us how it works? Is it the polarization, different polarizability of them? Or?
You mean the fact that they phase separate?
Just the motility pushing.
We think it's motility. So this is something that they are now working also in simulations to try to, because I mean, in principle, the cluster should break. I mean, what we think is also that the fast particles, I mean, the hydrodynamic flows are a bit stronger. So then for some reason, there is like also some like motility-induced phase separation here, although we are not in that phase. So we're trying to explain it theoretically because it's not very clear why this will phase separate. We were really far away from motility-induced phase separation. That's why. So yeah.
You can measure the flow fields.
Yeah, we can track them. Yeah.
But there are like some dipoles or force dipoles or whatever.
Yeah, exactly. So now we're trying to look into the details on that because it was not it was not necessarily intuitive why this has to happen for us. But yeah, that's a good question that we're trying now to solve.
Theories.
Yeah, exactly. Okay, so up to now, I can show you that material properties are important, and also the interaction with environments in terms of like light, stimuli, also number of particles, right? And that we can design motion. And that's basically what happened here with thermoresponsive light and also like external fields and a big number of particles. But come back to this picture. It's just a hard colloid, right? And my dream is really to do this, like this thingy. I want to do that experimentally and have this micro device that looks like a, well, that works, at least looks and works as white blood cell. So this is basically the effort that we're pushing now in my group. So trying to do this, let's see how long that takes. And so one of the things that we need to do, first of all, is leave behind hard colloidal particles. And this is very sad for me because I've been working with this for now 10 years. But of course, one has to evolve, right? So the idea is, yes, now try to look for architectures that look a bit more like a cell. And the first thing is just look for compartmentalized architectures with boundaries that, for example, can be deformable, like we can play with mechanical properties. These boundaries can also be semi-permeable or permeable to exchange information with environment. And of course, the thing that motivates me the most is what are the tools that we can use to make them motile. One example that I always put is also like, how can we, for example, reproduce the run and tumble motion pattern of bacteria? Maybe just in case you are not familiar with this, bacteria have this dynamical pattern when they just, where the flagella are aligned along the long axis, they can run directed to one direction. And time to time, and this depends on bacteria strain, they will stop by just reorienting the flagella. So they will have like no forces pushing, I mean, isotropically, so they will move. And they will smell what happens there, you know, and if they like it, they will stay there. But if they don't like it, they will come back to the running state and just go away from there, basically. So this is one of also the motivations to try to reproduce this motion. And so what is the experimental model that we're using to reproduce this? So there is many on the market, but we stick to giant unilamellar vesicles or lipid vesicles. So this, well, I don't need to explain you guys what it is. I probably, most of you know already. But basically, there's these compartments that are formed by self-assembled phospholipids, these amphiphilate molecules. And this is basically minimization of energy in water. And in principle, I mean, if you have this in aqua solution, so you have aqua solution outside, aqua solution inside. And it's very nice because you can play in a broad range of sizes, from the nano to the microscale. The mechanical properties are also tunable by playing with a phospholipid composition. And they're actually not that permeable, but you can play with the permeability by including, for example, proteins and different other biological units that will help with that. And so in the so I'm an active matter person, right? And it's been now since like 5 years that people are trying to really push in activity on these units. For example, in the UK, they have encapsulated bacterium that is able to basically from the inside push in this vesicle by coupling the flagellum motion with the membrane through this tube. And I have even tried to reproduce the active catalytic Janus particle with lipid vesicles by just coating one of the sides with enzymes, like we catalyze a chemical gradient, and this will move. Now, there is 2 problems with this. First, in this case, this depends on the bacteria time living, and also bacteria secrete a lot of surfactants and things that vesicles don't like. So at some point, this will probably explode. So it's not sustainable. And in this case, so enzymatic reactions, although I like them a lot and I'm start working with them, they are not very efficient, especially when they're attached to surfaces. So basically, in order for this particle to move in a directed way, it takes forever, and thermal noise mostly overcomes this type of motion. So we were trying to work on this a little bit and just, you know, see, can we actually make some, explore all the types of motion with Janus lipid vesicles and even reproduce run and tumble. And so this is basically the main work of my PhD, Vivian Willens. And she has done an amazing work so far. So we have this article in the archive in case that you wanna check it. So basically what she has done is to produce phase-separated lipid vesicles, Janus lipid vesicles, by mixing 3 types of lipids. I mean, the most important one are these 2 lipids that have a different ordering in the layer at room temperature, and they have a very nice phase separation. They overcome a phase separation at room temperature, like within hours. And so you end up with these very nice vesicles with two faces that are very stable in solution. Of course, the process that we use is a protocol called electroformation, and I will be lying to you if I will be showing you this picture. In fact, we get a variety of different shapes. We get some particles that, some vesicles are not fully phase separated. We get these dumbles where you can see that there is like line tension in between both faces, and they form this peanut-like shape. And we also have this kind of, weird shapes where you have kind of two faces separated by a membrane. But just for the sake of time, I will mainly focus in of the Janus vesicles. Of course, this is also very interesting when you want to play with geometry and motion, right? So these vesicles are usually between 5 to 10 micrometers diameter. And again, in this particular picture, I show you that the phase separation is 50-50. This is not always the case due to the type of protocol that we use for making them. We can also have like a different, so maybe 40, 60, or 30, 70, this phase separation. So, but I mean, that's okay. We're looking into dynamics, so that's fine. And so what we did now, it's okay, this is a new system. Let's try to not change too much what we are doing. So we have a new colloidal system membrane. Let's use electric fields again. Let's see what happens. Is anybody worried about lipid vesicles and electric fields in the room? I mean, you should be, because there is certain conditions on electric fields that basically you will destroy them. But yeah, so my student actually did a very nice job on trying to quantify what happens at different field conditions. So again, we have these two electrodes, a solution of vesicles that will sediment, because we basically change the density of the solution, and then we apply an electric field. And so Vivian characterized the behavior of these vesicles by changing the amplitude of the field and the frequency of the field. Again, we are always on the kilohertz range. So indeed, there is a frequency from 5 kilohertz a little bit down to 1. Actually, where usually active colloids are the fastest, where we usually work, where the vesicle breaks. And this is, let me see if I can play the video. So this is going to be very fast. So when we apply in this red regime, so the vesicle just basically explodes. I'll just play it again. And this is, so you have to imagine, or first, there's a couple of things happening here. At these frequencies, people use actually to create pores in the membrane, so what is called electroporation. But also what you have to imagine the membrane as a capacitor, right? So there's charges that accumulate in the membrane. You start building a very, very high tension in this membrane up to, the membrane cannot hold it anymore and just breaks. Then we have another regime, so this black, where nothing really happens at higher frequencies. And of course, you have to imagine this, again, we're calling about polarization timescales. So if your electric field is too fast that it's not able to polarize the charges around each hemisphere, it's virtually like doesn't exist. So nothing really happens, right? And then we have this very nice, we were very happy to retrieve this very nice blue region, which is the blue region of active motion. And we motivate this, and I will show you in a bit that we have actually active motion by induced charge electroosmosis. And when it happens close to the electrode, then we have these hydrodynamic flows that will move the particle as it happens for the Janus particles. And so I will just show you a video. So this is how the variety of vesicles that we have. And when we switch on the field, you can see the different sizes. I mean, they move differently, different geometries. But for example, these are kind of the ones that my student pick for the statistics, right? Very patient for that. So you can see that they move around, right? They don't break, they kind of move around. We can characterize dynamics. And so, yeah, I mean, that's what we start doing. And so one of the things that we know is this happens again, because first of all, the lipids that we use, so it's the OPC, the PPC, and cholesterol, they are not charged. But we use dyes that are charged, and the amount of dyes in each side is different. So you will have a different surface charge. And therefore, again, we come back to this idea of hydrodynamic flows that depends on the surface property, the electric surface properties. And we know by PIV that the flow magnitude and direction is more important on the green side, and these particles will move with the red side in front, okay? So we were very happy to find that out. And then we started quantifying dynamics as we do for active particles. So basically, we measure them quadratic mean displacement, so the mean square displacement as a function of time. And we measure this for different fields. field conditions. If you remember, I told you that the velocity of the particle, it depends on this field strength, so higher field, more velocity, so that's what we see. And then usually for active particles, when we measure the MSD, we have always a transition from this ballistic state to diffusive state at a time scale that depends on the reorientation of the particle. Because basically what happens is that if you see this trajectory along scale, this particle will be randomized and will look actually like a Brownian trajectory, so a diffusive trajectory, okay? Now, rotational diffusivity should not be influenced by the field, because as I said before, it depends only on thermal fluctuations inside. But in fact, what we saw when we measured this is that when we increase our electric field voltage, basically, we did have a decrease of this reorientation time. And at the beginning, I was very worried. I was like, okay, there's something we're doing wrong. Just do the experiments again, Vivian. She did it again, but it was consistent. It was always, there's this decrease of on time, reorientation time. And so we wanted to compare what will be the expected theoretical reorientation time for this particle. So what you see here is basically, so the orientation time is 1 over the rotational diffusivity. And so we measured this theoretical reorientation time for particles of about 10 micrometers diameter. And then we plot that with our reorientation times for our, what we measure, basically the experimental. You can see that there is like a tendency to so the reorientation time becomes faster when we increase the voltage. So we basically found an extra time scale that we were not expecting, but I'm gonna show you why. So I will play again the movie, but just focusing on one particle. And now, so you do have active motion pushing towards this side. I mean, you can even see the tracers that are there for tracking the flows. But if you take the example of a distinct hard sphere, where you have 2 sides, yeah? Yeah, so now lipid membranes are fluid. You don't have 2 discrete hemispheres. And time to time, I mean, actually all the time, this fluctuates quite a lot. And I don't know if you saw it, I'm just gonna play it again, but there is a specific moment, so this starts swimming, and it will happen over here. So it fluctuates quite a lot, so there's quite a noise when you measure the velocity. It's quite noisy, but still you have this moment where it just runs. So, I already spoiled it, but anyways, it just goes fast. And then it will happen over here that at some point, and this is still an open question, what happens, at some point, the whole phase separation will disappear from asymmetric to symmetric here, mixes together, and then just phase separate again, kind of, and keep swimming, right? And so it was very fun to look at this. And what we tried to explain, or like the explanation that we gave it behind, is that you have this transient dynamical states between what we call a run and a tumble state. Running meaning when the particle, it's completely fully phase separated and it runs, so the velocity will be maximum, okay, maximized. And then you have this moment where we believe are interactions with the bottom electrode, because you have to imagine the surface as a rough surface. Although it could be also hydrodynamic effects on flows around the particle, inside the particle, but this is an open question right now. But anyway, so what happens is that time to time, this phase separation mixes, and now you have loss of asymmetry, so the particle doesn't have, it doesn't move, it doesn't propel, right? And so
geometrical effect that they are up or down?
No, you actually see that. So it's actually very complicated, because we try to measure with confocal microscope, but because they're moving and our confocal microscope is not like the best in the market, it's very hard to see. But there is a couple of papers by Dimova where they actually see that this happens in 3D when you have an electric field applied. So it's not only that they rotate, it's really due to the fluidity of the membrane and now that lipids can mix. So the energy barrier for this mixing is actually low enough that for any hydrodynamics or interaction with a solid substrate can affect that. And so what we did to, make a strong point that this is actually run and tumble-like behavior, we use exactly the same analysis that people use for bacteria. So what people usually do is to measure the velocity and the reorientation, or like the direction basically, as a function of time. And usually if you have to imagine this tumbling effect, like the particle stops, so the velocity drops and it becomes, it starts jiggling quite fast in place. So the direction will be like this difference in between 2 orientations will be very fast. And when it runs, the velocity is maximum, and this reorientation will be much, much slower. So we do that actually, and what we found, so all the gray lines are basically the overlap between a drop in local velocity and an increase on this orientation. And we were very happy to see that when we find all of these gray lines, it exactly coincides with what we call this tumble state. Something we can do also to make a stronger point is to measure what we call a order parameter. So this order parameter is a bit, a bit fishy actually, because basically what we're measuring here, it's so our order parameter will be maximized, so it will be 1 when looking at the equator of our particle, you will have 2 phases. And it will be minimized when you have like disorder state. So basically green and red are all mixed right together. In principle, what we say is that if you have a higher order parameter, basically you will have phase separation that is very distinct, so the velocity will be higher. If you have a lower order parameter, it should coincide with the tumbles, and it does. However, you see that this is not always decreasing, even there is a lot of tumbles, because we're measuring just in one plane. So there is also a projection in 3D that we are actually overestimating our order parameter. But at this moment, we don't have better ideas for this because, of course, again, with confocal imaging is very complicated because the particles are constantly moving. But I guess it gives a good approximation of at least for the runs, completely runs and completely tumbles in this state. And so this is very nice because, of course, now we have an extra time scale that comes from the tumbling. And I didn't say it, but this phase separation depends a lot on temperature. So this is a temperature-driven phase separation. So one of the things that we would like to explore is basically, okay, now if you have temperature gradient, for example, in our sample, can we actually control where this mixes or not mixes by changing temperature? And just to finalize, I don't know if it's fine, okay. So just to finalize, I want just to showcase to you many of the other very interesting things that we can do with these vesicles. Of course, they're now hollow, so as cells, we can start putting things inside, and this is something that people have done. I mean, in our synthetic biology, they're doing quite a lot, like trying to charge them with different biological materials. But what I want to just show here is that we have a very nice accident while producing these vesicles, where we have a vesicle inside of a vesicle. And the big vesicle, it's able still to move around with a small cargo inside. So, I mean, in principle, we could start encapsulating other things here. For example, we have rods. So the red things are just rod particles, and this green is just like some extra material from the membranes or lipids, basically. And now we're trying to explore also the encapsulation of other type of colloids, molecules, and see if we can just basically direct this type of systems towards one direction. And if you remember, I told you that we can also break the vesicle. So imagine that now we have achieved, we are somewhere in the space that we want with our cargo. We can just make the particle burst and the vesicle burst and release the cargo that, as you can see, is untouched by this change in electric field. So this is, again, this electroporation and membrane tension effect that will break the particle. And finally, this is experiments that my student got to me recently. We can also start to look at division. So now we have a multilamellar vesicle. So this is like an onion, let's say, that has many, many vesicles. So we apply an electric field. And I hope you can see that it kind of unfolds when you apply this electric field. And it tries to, something weird is happening. We don't know what's happening, but yeah, beautiful things are happening. And then there is this small vesicle that just departs from the main vesicle. What we think is happening here, of course, membrane tension effects and the fact that you have extra material. So usually when you apply an electric field, the vesicles shrink a bit. Here you have an onion with extra material that appears. But we believe that we are actually doing electroformation in 3D. So this is basically, electroformation works as you have a lipid layers on a substrate, you apply an electric field, and you have also an aqua solution, and then we hydrate with this electric field. So we believe that we have something similar, where this is very nice, because now with microfluidics, we could actually choose the composition in each layer of the onion, and try to have deliver of these different compositions by division. And actually, division is quite of a big question in synthetic biology at the moment. So yeah, I mean, this is more or less everything I wanted to tell you. I hope that I convinced you that even if colloidal particles are great, vesicles are better, especially for some of the, for some of the ambitions that some people here in the room we have. I hope that I show you that we can indeed make them motile, that because of the very distinct material properties of bilipid layers, we can also play with the motility pattern. And now we can also play with compartmentalization sterilization, and cargo delivery. And some of the things that we are also exploring right now, as I told you before, is how this behaves in in temperature gradients, because this phase separation will, at higher temperatures, this will mix. So instead of just having like random mixing, we will actually spatially control it. And we're also working a little bit on encapsulating active particles with microfluidics in membranes. And this is also a very preliminary video of active particles actuated with electric fields. So the membrane now is completely homogeneous. There is no phase separation. And I hope that you can see there is some sort of beating of this. We don't know what's happening here, but I'm just showing to you just in case you have any ideas, Antonio. And then this is slight particle here, attaches spins to the membrane and stays there. And this video is a bit long and slow, but at some point, this guy will start moving into the other direction. So those are the things that we are trying now to explore. And yeah, at some point, we will drop behind the electric fields and work with more chemically active particles because, electric fields are still far away from biology. And as a very last slide, so this is a collaboration that's happened very recently with my colleagues in the German Aerospatial Institute. So we're sending microgravity experiments with our lipid vesicles because apparently, and I don't know why, and this is something that's why we're doing it, fluidity is affected by microgravity. So there's a couple of papers published on nano vesicles that have been sent to microgravity in the sounding rocket, where they measure fluidity changes in, yeah, that was my face too. They actually measure fluidity changes in these nano vesicles. So what we're doing now is basically do the same experiments but now with micron-sized vesicles, or like the same vesicles that I showed you. And so this is a collaboration with the Swedish Space Corporation and DLR, especially Thomas Voigtman and Ivo Butinoni. And I was there, this is our rocket. I was there 1 month ago to basically launch these vesicles to microgravity, sorry. If you have questions on any ideas on this, now I'm recruiting people that will be interested on this type of experiments. And yeah, so with this, I would like to thank first of all of you, of course, everybody that make possible the work that I have to show you, all the funding agencies. And I didn't put it here, but so we just kick off doctoral network, Marie Curie, like the older ITNs on synthetic biology. So it's a SYN6L. So we are recruiting 12 PhDs, different types of projects, in case that anybody's interested. And yeah, with this, I would like to thanks for your attention, and I'll be glad to take more questions.